I'm Eunice, a branch chief in the Division of Molecular Genetics and Pathology within the Office of In Vitro Diagnostics and Radiological Health in the Center for Devices and Radiological Health at the FDA. Today, I'll talk about understanding how to use databases in the assessment of the clinical validity of variants, the individual technologies, and their error rates. As part of President Obama's Precision Medicine Initiative, the FDA is working toward optimizing regulatory oversight of NGS-based tests. To this aim, we are interested in supporting and participating in regulatory science research in order to gain a deep understanding of the performance and validation of NGS tests, as well as to understand how the test results are interpreted and communicated. In April of this year, the UCSF-Stanford Center for Excellence in Regulatory Science and Innovation, or CERSI, and the FDA co-authored a perspective in Science Translation Medicine, in which a research roadmap was proposed with the following 9 areas of potential research. The first 5 areas address aspects related to the analytical performance of NGS tests, and the next 4 are related to the clinical performance. Today, I'll discuss the 8th topic about the development of methods for using public databases for regulatory decision-making. NGS tests are rapidly being adopted in the research and clinical settings. This is leading to a rise in the identification of human genetic variants, including rare variants, at an unprecedented rate. Variant information, including the evidence supporting the variants, may be collected and stored in databases where the data can be annotated, curated, and interpreted for clinical significance. For example, for the identification of clinical genotype-phenotype relationships. Such databases may be made publicly available in order to serve as public resources for understanding functionally critical variants with clinical significance. A few examples of freely accessible databases that currently catalog human genetic variants include ClinVar and ClinGen, PharmGKB, and the Human Gene Mutation Database. There are also locus-specific and disease-specific public databases, such as the Clinical and Functional Translation of CFTR, or CFTR2, and the Breast Cancer Information Core, or BIC. And these are just to name a couple. As variant interpretation is dependent on the data that's available at the time of classification, information within databases is typically collected and considered over time according to defined procedures and processes. The procedures by which a database is monitored, maintained, and updated, the curation process, and the evidence used to support conclusions about the clinical relevance of a variant are all key to evaluate the validity of the conclusions in the database. Thus, FDA is interested in devising strategies to evaluate and understand these aspects so that publicly accessible databases may serve as a source of valid scientific evidence to support the clinical validity of the variants that are detected by NGS tests. When a device is considered for marketing authorization, FDA relies upon only valid scientific evidence to determine whether there is reasonable assurance that a device is safe and effective for its intended use. During pre-market review, the performance of an in vitro diagnostic device, or IVD, is based on analytical as well as clinical validation. Analytical validation addresses whether a test measures the intended analyte or analytes accurately and reproducibly. Clinical validation addresses whether the test result is correlated with the condition of interest in a clinically significant way. Public databases that curate and interpret human genetic variants are one form of evidence that could be considered to establish clinical validity of the variants that are detected by NGS tests. In fact, this has been done before. In 2 cases, locus-specific databases were reviewed along with other validation data in order to support marketing authorization of 3 sequencing-based devices that were reviewed in the Division of Molecular Genetics and Pathology. I'll discuss these 2 cases to describe how the different databases were reviewed in order to support the clearances and approval of the devices. In the first case, the CFTR2 database was used to establish the clinical validity of the Illumina MiSeq Dx CF assays. The Illumina MiSeq Dx CF139 variant assay and the Illumina MiSeq Dx cystic fibrosis clinical sequencing assay are NGS-based tests that were cleared in 2013 for the detection of variants in the CFTR gene. Previous clearances for other CF assays use published literature references in order to establish clinical validity. However, this approach was not feasible or practical, considering the relatively large number of variants that are detected by the 2 NGS tests. So the CFTR2 public database was used. CFTR2 is a global effort between CF registries, clinics, and researchers focused on the identification and classification of CF variants in patients. Variants are characterized as CF-causing, non-CF-causing, mutation of varying clinical consequence, and mutation of unknown significance. All of these classifications are based on defined criteria. For the CF139 variant assay, only variants established as CF-causing, with the exception of 8 variants, were included in the assay. In contrast, the CF clinical sequencing assay is indicated for patients for whom available CF panels failed to detect CF-causing variants. So variants that are not covered by the panel assays, such as novel variants and rare variants that occur at very low frequencies, could be detected with this test. As a result, the labeling indicates that the test results should be interpreted by a board-certified clinical molecular geneticist and that another sequencing method should be used to validate any novel variants. In the second case, a proprietary database was evaluated as part of the device. However, it was not used to establish clinical validity. In part, this was due to the intended use of the device. In 2014, Myriad's BRCA-analysis CDX was approved as a companion diagnostic for the drug olaparib. Therefore, the clinical validity of the test was based on results from the clinical trial that was used to support drug approval. This test utilizes Sanger sequencing and multiplex PCR to detect variants in the BRCA1 and BRCA2 genes. Notably, the classification process was approved as part of the device. As with the CFTR2 database, defined criteria are used to classify variants into a range of pathological categories, of which 2, deleterious and suspected deleterious, are eligible for treatment with olaparib. Since Myriad's database is not public and all variants were not represented in the primary efficacy population, and variant classification could change as evidence becomes available, the device was approved with a condition to provide post-approval updates on the variant database so that, in part, trends in variant classification changes could be tracked. Although the database did not establish clinical validity for the variants detected by the BRCA-analysis CDX test, the database was approved as part of the test to allow for the variant class claim for this device. So taken together, the 2 case studies that I presented may serve as the foundation for developing recommendations and methodologies for reviewing and recognizing public databases to support device performance. Specifically, leveraging public databases for regulatory science may facilitate development of ways to evaluate and understand controlled processes for data management, curation, and interpretation. and also for ways to recognize the databases as resources for translating genetic information into useful clinical evidence. Ultimately, relying on assertions in publicly accessible human genetic variant databases for regulatory purposes could reduce regulatory burden on test developers, and it could encourage advancements in the interpretation and implementation of precision medicine. If you'd like more information about the case studies that I discussed, the links to the decision summaries for the CF assays and the summary of safety and effectiveness data, or SSED, for the BRCA-analysis CDX test are listed here. If you have any questions about the information presented today, please feel free to send me a message, or please send a message to the PMI mailbox. Thank you very much.